which can be accessed anytime, anywhere through recorded sessions. Then apart from that, we have notes and MCQs based on the latest syllabus so that you can assess yourself. Then apart from the academic guidance, we have a very special thing about Auricle, which is we also provide personalized counseling sessions. So anytime you can book a slot with us in case you need that, you want some extra support and guidance from our end, apart from the academic support that we're already providing. Then we have 3 different subscription plans at Oracle, starting from the basic, which is for 3 months and will cost you 15,000. Then we have the standard plan, which is for 6 months and is available for 30,000. Then we have the premium plan, which is for 12 months and will cost you 50,000. In the premium plan, we have live doubt clarification sessions and also one-to-one personalized guidance. Then you can also use my promo code as displayed here on the screen to get an additional 20% discount on all the subscriptions. Then in telling you a little about the internal exam marathon series, this series has been specifically created for your university exams. So we have rapid revision of all important topics taking place in this series in a short span of time. We are trying to touch upon all the important topics, at least superficially, and tell you that what is the most frequently asked questions. Then volatile facts will be quickly revised. Multiple choice questions will be covered so that we can assure you top-notch grades in your exam. Okay. So beginning with urea cycle, but before that, I'd like to talk about the toxicity of ammonia. As I said in the previous lecture, that ammonia is very toxic. So why is all this handling of ammonia so much required and in such a regulated way? Because ammonia is very toxic for the body. So before we begin with urea cycle, that how that ammonia is ultimately removed from our body, let's understand the consequences. What if this ammonia somehow accumulates in the body? Okay, so we'll be discussing about the toxicity of ammonia. So in blood, if there is increased ammonia, it usually combines with hydrogen ion and is transported as ammonium ion. So in case of little amount of ammonia, if there's little amount of ammonia in the blood, what will happen? It will combine with hydrogen and form ammonium ion. See, in the blood, if there is some little amount of ammonia, okay, what will happen? It will combine with hydrogen ion to form ammonium ion. Okay, so this is how it is transported. Now, this ammonium ion, it is impermeable to the blood-brain barrier. To blood-brain barrier. So that is in case of little amount of ammonia in your blood. So it is transported as ammonium ion, which is impermeable to the blood-brain barrier. Everything sounds okay. But what happens in case of excess ammonia? Okay, now when there is excess of ammonia in the blood, all of it cannot be converted to ammonium ion. Okay, it will lead to alkalosis. So what happens? The ammonia accumulates in the blood. And now this ammonia is able to cross the blood-brain barrier. So this ammonia, in case of excess ammonia, this ammonia, all of it cannot be converted into ammonium ion and it will lead to alkalosis. So it accumulates in the blood. Now this ammonia, it is able to cross the blood-brain barrier. Now in the brain, what happens? This ammonia is taken up by alpha ketoglutarate to form glutamate, which in turn form glutamine. So what is happening? That ammonia, which is able to cross the blood-brain barrier, it is going to enter the brain and then through transamination, it will combine with alpha ketoglutarate to form glutamate. And in turn, that glutamate will again accept ammonia and form what? Glutamine. Now, this glutamine, it is osmotically active compound. That is, it has the property, it is able to attract water molecules causing cerebral edema. So you see how these are the toxic effect of ammonia. Also, what happens that glutamate that is formed, it on decarboxylation will produce GABA. Now, what is GABA? It is an inhibitory neurotransmitter and it leads to CNS depression. Okay, so you already studied that how your amino acids on decarboxylation they produce amine. So GABA is an amine, and when glutamate is decarboxylated, it produces GABA. Now, this GABA is an inhibitory neurotransmitter and it causes CNS depression. Also, what is happening, you see, your alpha ketoglutarate is being depleted, okay? And alpha ketoglutarate is an important substrate for TCA. So what is happening? Your TCA is getting affected and there is decreased ATP production. Now, ATP production is being decreased because of your TCA being affected, and also when glutamate is changing into glutamine, we know that the glutamine synthetase enzyme, it also requires ATP. Now, there also your ATP consumption has increased and ATP production has decreased. So what is happening? Overall, the ATP production has decreased. So these are the side effects or the toxicity of ammonia. So if we recapitulate what is happening, because okay, now this ammonia, it is able to cross the blood-brain barrier and it reaches the brain. Now what happens? It combines with alpha ketoglutarate. So what happens? Your alpha ketoglutarate decreases. So your TCA, that is TCA cycle is affected, and ultimately what is happening? Your ATP production is decreasing. Also, what is happening when alpha ketoglutarate is combining with ammonia? What is being formed? Glutamate is formed. So your glutamate levels are increasing. When glutamate increases, what happens? It changes into glutamine. So your glutamine level is increasing. And glutamine, as I said, it is an osmotically active compound. So what happens? It is able to attract water, leading to cerebral edema. Also, glutamate on decarboxylation, it produces GABA. Now, what is GABA? It is an inhibitory neurotransmitter. So it causes what? It causes CNS depression. Okay. And what is the full form of GABA? It is a biologically important amine and it stands for GABA aminobutyric acid. Okay. So it is the primary inhibitory neurotransmitter in the brain. So GABA is your GABA aminobutyric acid. Okay. So that was briefly about the toxicity of ammonia. Okay. Now beginning with urea cycle. So urea cycle is also known as Krebs-Henseleit cycle. It is also known as ornithine cycle. Ornithine cycle, why? Because your ornithine is regenerated. Ornithine is regenerated. I think there is some audio video lag anyway. And it is also known as urea bicycle. Now, why bicycle? Because your TCA and urea cycle, they are linked together. Because TCA and urea cycle, they are linked together. Let me show you how. So, in TCA, what is happening? The aspartate that is formed is entering what? It is entering urea cycle. And in urea cycle, the fumarate that is formed is entering your TCA. So this is how your TCA and urea cycle, they are linked together and therefore also known as urea bicycle. Okay. Now introducing you to the structure of urea. So as you can see in the picture that I have shown, that there are 2 nitrogens and 1 carbon. So your urea consists of 2 nitrogen and 1 carbon. So one of the nitrogens, it is derived from ammonium ion. Okay, this and the other nitrogen, it is coming from aspartate. And this carbon is from respiratory carbon dioxide. Okay. Now, site, your urea cycle takes place only in the liver, and organelles is it is both cytoplasmic and a mitochondrial cycle. And we say cycle and not pathway because your ornithine is regenerated. Now moving on to the reactions of urea cycle. So we see in the first step, these boxes in pink here, this and this. These are indicating what? That these reactions are taking place inside the mitochondria, okay? Okay. So what is happening in the mitochondria? Your NH3 and carbon dioxide, that is your ammonia and carbon dioxide, they're combining to form what? Carbomoyl phosphate. So the first step is formation of carbomoyl phosphate, and this is brought about by the enzyme carbomoyl phosphate synthetase. Okay, and this is the rate limiting enzyme. This is the rate limiting enzyme. So what is happening? Ammonia, it combines with carbon dioxide to form carbomoyl phosphate. This is taking place in the mitochondria, and what is needed, as you can see, 2 ATPs are being changed into 2 ADP. Okay, so 2 high energy phosphates are needed. And also for this step, NAD is an obligate allosteric activator. It is an obligate allosteric activator. That is, this reaction will take place only in the presence of NAG. And what is NAG? It is N-acetyl glutamate. Okay. Now the second step is that your carbomoyl phosphate, it combines with ornithine to form citrulline. And this is brought about by the enzyme ornithine transcarbamylase. Okay. Ornithine transcarbamylase is bringing about this reaction. So your carbomoyl phosphate combines with ornithine to form citrulline. This is also taking place in the mitochondria. Now this citrulline comes out into the cytoplasm and it combines with aspartic acid. Okay. Now this aspartic acid, it is coming from the TCM. Now your citrulline, it combines with aspartic acid to form arginosuccinate. The enzyme is arginosuccinate synthetase. And for this reaction also, two high energy phosphates are required, but 1 ATP, which is equivalent to 2 high energy phosphates. Why? As you can see, 1 ATP is changing into AMP, that is a triphosphate, adenosine triphosphate changing into adenosine monophosphate. Okay, and this aspartic acid, as I said, it comes from TCA. It comes from TCA. So, and your TCA, it is taking place in the mitochondria. TCA takes place in mitochondria. So what happens? Aspartate comes via a transporter which is known as citrin transporter from the mitochondria to the cytoplasm. So your aspartate via citrin transporter comes out from mitochondria to cytoplasm. Okay. Now what is happening? Your arginosuccinate that is formed, it is acted upon by the enzyme arginosuccinate lyase. And what is formed? Arginine and fumaric acid is formed. Now this fumaric acid will again enter TCA. And this arginosuccinate synthetase, it is a lyase enzyme. So arginosuccinate lyase, okay? It is in lyase enzyme, as the name says itself. So what are lyase enzymes? They are involved in breaking covalent bonds without adding water. So breaks covalent bond without adding water. Now what is happening? Your arginine that is formed, it is acted upon by the enzyme arginase, and this arginase is a hydrolase. Arginase is a hydrolase. So this hydrolase, it breaks covalent bond. It breaks covalent bond by adding water, as you can see here. So what is formed? Your arginine acted upon by arginase, and what is formed? It splits into urea and ornithine. So your ornithine is regenerated, and this urea you can see here, this urea has been formed. Now this ornithine that has been regenerated, it again from the cytoplasm goes to the mitochondria and again combines with another carboxyl phosphate, and the cycle continues. So these were about the reactions of urea cycle. Very important, okay? Hardly any reactions, 3 to 4 reactions, very simple to remember. The initial 2 steps are taking place in the mitochondria, the rest of the steps are taking place in the cytoplasm. Okay. Moving on to the energetics of urea cycle. So your CPS1 uses 2 ATP, which is equivalent to 2 high energy phosphates. That is your carboxyl phosphate synthetase 1. That is the enzyme, first enzyme of the urea cycle and takes place in the mitochondria. It uses 2 high energy phosphate. In arginosuccinate synthetase, it uses 1 ATP but 2 high energy phosphates. So the net is that 3 ATPs are directly used, but which is equivalent to 4 high energy phosphates being used in urea cycle. Now moving on to the regulation. High protein diet increases urea cycle enzyme synthesis, which is very important. If you're taking more protein, so there will be more nitrogen. So there will be more urea cycle operating. So urea cycle enzyme synthesis will be increased. NAG is an allosteric activator of CPS1 and it is an obligate allosteric activator. That is, without the presence of NAG, the first step will not take place. And compartmentalization, as we've already seen that the first and second reactions, they're taking place in the mitochondria and the rest of the reactions, they're taking place in the cytoplasm. So this compartmentalization also provides regulation. Now moving on to the disorders of urea cycle, very important. Okay, so if there is any defect or block in the first enzyme, that is carboxyl phosphate synthetase, so it will lead to hyperammonemia 1, type 1, hyperammonemia type 1. Now, if the block is in the second enzyme, that is ornithine transcarbamylase. I think there's some typing error in this diagram. Ornithine transcarbamylase. Okay. Okay. So, if the defect is in this second enzyme, what will be caused? Hyperammonemia type 2. Okay. Now, these two reactions, they were taking place, as we know, they were taking place in the mitochondria. So I've separated this. Okay. Now what happens? This citrulline that is formed, it combines with aspartic acid, aspartate, and it forms arginosuccinate. Okay. Now what happens? If there is some defect in the transporter, okay, in the citrin transporter, what will happen? It will lead to citrullinemia type 2. So if there is a defect in citrin transporter here, that is your aspartic acid, it is not able to come out from the mitochondria into the cytoplasm. That is the case, okay? So what will happen? Your citrulline will accumulate and this is citrullinemia type 2. Type 2. So when I'm saying type 2, there must be a type 1. So type 1 is caused by arginosuccinate synthetase. Okay. This is synthetase. Okay. So this causes, that is your arginosuccinate will not be formed. That is, citrulline and aspartic acid will not be able to combine and form arginosuccinate. So again, what will accumulate? Your citrulline will accumulate and will be citrullinemia type 1. Type 1. Okay, now your arginosuccinate, it was acted upon by the enzyme arginosuccinate lyase to form arginine. Too many spelling errors in this diagram. Okay. So your arginosuccinate lyase, if there's some defect with this enzyme, it will lead to what? It will lead to arginosuccinic acid urea. Arginosuccinic acid urea. Okay. Now, if there's some defect in this arginase enzyme, which is the last enzyme, what will accumulate? Your arginine will accumulate. So it will cause arginemia. So you see, if there's defect in the initial two enzymes, then what is accumulating? More of ammonia is accumulating. So it will cause hyperammonemia. But you see, later on, if there is block in some enzymes that are coming later on in the cycle, it will lead to the accumulation of the intermediate. So the disorder is named on the accumulation of the intermediate that is being accumulated. Okay. Now, there is this diseases and enzyme deficit in the form tabular form. So I've already taken up in the diagram. It is more easy for you to remember that way, but I'll just repeat it in the tabular form as well. So your hyperammonemia type 1, it was because of the first enzyme, that is CPS1. Hyperammonemia type 2, it was because of ornithine transcarbamylase. Okay. Then citrullinemia, I discussed two types. One was because of arginosuccinate synthetase. Excuse me. And the other was because of defective citrin transporter. Then arginosuccinic aciduria, it was because of arginosuccinate lyase. And hyperarginemia, it was because of arginase. Now there's another hyperornithinemia. This is here. That is when your ornithine accumulates, and it is because of the defect in the transporter that transports your ornithine from the cytoplasm to the mitochondria. So defective ornithine transporter, and it is also known as HHH syndrome. HHH syndrome. Why is it known so? I'll just talk about it later in the lecture. Okay. Now moving forward to, we'll discuss some of the important disorders of urea cycle. So beginning with type 2 hyperammonemia. So here, what is the enzyme defective? That is your OTC, ornithine transcarbamylase is defective, leading to accumulation of carbamoyl phosphate. So the first step is happening, the second step is defective. So your carbamoyl phosphate, it accumulates. Carbamoyl phosphate comes out into the cytoplasm from the mitochondria and enters pyrimidine synthesis. Now, orotate is an important intermediate in pyridine synthesis. Okay. Which forms uracil and from which cytosine and thymine are obtained. So what is happening? Your carbamoyl phosphate, it comes out into the cytoplasm and enters your pyrimidine synthesis, and ultimately what is formed? Your pyrimidines, that is cytosine and thymine, they are formed. Now, an important intermediate in pyrimidine synthesis is your orotate. Okay, so orotic acid, which is insoluble in water, it forms orotate crystals, causing orotic aciduria. Now, very important that your orotic aciduria is a clinical manifestation of type 2 hyperammonemia. It forms pink gravels in the urine. Okay. So, I'll just explain you this. Just a second, my pen is not working. Okay. Yeah. So what is happening? Your first step is taking place. Here. Ammonia, it combines with carbon dioxide, and what is formed? Carbonyl phosphate is formed. Now, what is happening? This carbonyl phosphate, it was supposed to combine with ornithine to form what? To form citrulline. Now, this is not taking place, okay? This reaction here is not taking place. So, what will happen? The carbonyl phosphate starts to accumulate. So, this reaction, these are the first two steps. So, this was taking place in the mitochondria. Now, this carbonyl phosphate, which accumulates, it comes out from the mitochondria into the cytoplasm. Now, this carbonyl phosphate, it enters pyrimidine synthesis. And an important intermediate of pyrimidine synthesis is your orotate. So this orotate forms uracil, and ultimately what is formed from this uracil? Your cytosine and thymine are formed. So pyrimidines are increased. And also this orotate, it is insoluble in water and forms orotate crystals, leading to orotic aciduria. Insoluble in water. Okay. So what are the distinguishing factors of hyperammonemia type 2? We talk about them. Distinguishing factors for hyperammonemia type 2. The first one is orotic aciduria, and the second one is excretion of pyrimidine in urine. Urine. Okay. Now moving on to arganosuccinic aciduria. Here the clinical manifestation is trichorexis nodosa. That is, you have dry, brittle hair. So you can see okay, my pen is finally not working. Okay, so you can see small nodes. They are present in the hair. Now, HHH syndrome, it is due to defect in ornithine transporter. As we've already discussed, that ornithine that is regenerated in the cytoplasm, it via the transporter from the cytoplasm goes back into the mitochondria. Now, in HHH syndrome, there is defect in ornithine transporter. Now, clinical manifestations include hyperammonemia, hyperornithemia, and homocitrullinemia or homocitrullinuria. Now, as ornithine is not transported back from cytoplasm to the mitochondria, therefore, in the mitochondria, carbonyl phosphate combines with another basic amino acid, lysine, to form homocitrulline. So, what is happening? Your ornithine, it was being transported back from the cytoplasm into the mitochondria, and in the mitochondria, it was combining with carbonyl phosphate to form citrulline. Now, as the transporter is defective and ornithine cannot go back into the mitochondria, so what is happening? So, carbonyl phosphate in the mitochondria, it is combining with another amino acid, which is lysine. So, carbonyl phosphate, it combines with lysine to form homocitrulline. Okay. So therefore, you can see homocitrullinemia in case of HHH syndrome. Now moving on to arginemia. Here the enzyme defective is arginase, and it is the least hyperammonemia is seen in arginemia. Why? Because arginase is the last enzyme. So by the time arginine accumulates, both the nitrogen are already abducted. That is, they are in the non-toxic form. So what happens? which can now be excreted in the urine. They've already, both the nitrogen, they've already been abducted. They are in the non-toxic form and it can be excreted. Therefore, it is known as least hyperammonemia. So that was about urea cycle and the main common clinical features of urea cycle disorders. There's a classical triad, that is your encephalopathy, your hyperammonemia, and respiratory alkalosis. Okay, so your encephalopathy is actually, it is a broad term for any brain disorder that alters the brain function and structure, and we've already seen how in case when we were talking about ammonia toxicity, that in case of excess ammonia, how it is able to cross the blood-brain barrier and damage the brain. That was one thing. Then hyperammonemia, we've seen how. Then respiratory alkalosis. Because of excess ammonia, there is hyperventilation. So there is carbon dioxide washout is taking place because of hyperventilation. So in urea cycle disorders, you have tachypnea, that is fast breathing. So that was about respiratory alkalosis. So these are the classical triads of urea cycle disorder. So with this, we come to an end of our urea cycle disorders. Okay. Now I'll also begin with aromatic amino acids today because protein is a very vast unit. So we take the revision quickly so that we are at least able to touch upon maximum topics. So from aromatic amino acids, the most important here is its clinical. That is the diseases that we'll be studying like phenylketonuria and alkaptonuria and all that. Okay, tyrosinemia. So that is most important here. Sometimes you also get asked about like its anabolic fates, what are the products that are formed from it. So that is also uh, as sometimes, but not as important as your clinical. Okay, so beginning with amino acid metabolism, we'll be taking amino acid metabolism under three headings, that is their catabolic fate, their anabolic fate, and their synthesis. Like in case they are non-essential amino acids, that is they need to be synthesized in the body and they're not supplemented through diet. Okay, so today we'll be talking about phenylalanine and tyrosine. Now, phenylalanine, it is an essential amino acid. That is, it is supplemented through diet. It is non-polar and it is both ketogenic and glucogenic. So, if you have a look at the structure, just a second. Okay, if you have a look at the structure, this here is the alanine and this here is the phenyl ring. Okay, so phenylalanine. Now moving on to tyrosine structure, if we compare it, it is non-essential. Why do we say non-essential? Because it is synthesized in the body, and we will see that your tyrosine, it is actually synthesized from phenylalanine, and it is non-polar. But because of this hydroxyl group, as you can see here, this hydroxyl group, because it has a phenol ring. Okay, this is the phenol ring. Okay, so because of this phenol ring, there is a hydroxyl group. So this hydroxyl group is giving it some polarity. Therefore, it is the least non-polar among aromatic non-polar amino acids. And it is also both ketogenic and glucogenic. So comparing the structures of phenylalanine and tyrosine, the phenyl ring attached to your alanine, and in tyrosine there's a phenol ring. And what is the difference between phenyl and phenol ring? That your phenol ring, they have a hydroxyl group, as you can see here at this position. Okay. Now moving on to the metabolism of phenylalanine. Now your phenylalanine, it converts to tyrosine. Now, the catabolic fate of tyrosine is that it is both ketogenic and glucogenic, and anabolic fate is that it is involved in the synthesis of menalin, catecholamines, and thyroxine. Okay. pen still not working. Anyway, there's not much to write. Okay, so talking about the metabolism, if we discuss about the catabolic trait, it is both your ketogenic and glucogenic amino acid. And talking about the anabolic trait, it is involved in the synthesis of melanin, catecholamines, and thyroxine. Now we'll take each of this one by one. That is its catabolic trait and its anabolic trait. But before that, there is the statement that your phenylalanine and tyrosine, they are twin amino acids. Explain why. So you might get this in exams. So it is because your phenylalanine cannot enter catabolic or anabolic pathways unless and until it is changed into tyrosine. So it can perform its functions only by being converted to tyrosine. So the functionality of phenylalanine is taking place only when it is being converted to tyrosine. In a way, it is functional only if it is converted to tyrosine. Now this reaction is taking place in one direction. That is your phenylalanine, it can convert into tyrosine. But your tyrosine, it cannot convert back into phenylalanine. So it is taking place in the one direction. So we say it is acting as a valve. Okay. That is, there is only forward side to this reaction. Now this reaction itself is very important. That is conversion of phenylalanine to tyrosine. It is very important. And a very important clinical is associated with this, which makes it all the more important. Now, this conversion, as we've already discussed, it is an irreversible reaction. That is, phenylalanine can convert into tyrosine, but tyrosine cannot convert into phenylalanine. Now, phenylalanine hydroxylase is a oxygenase. These are those enzymes that catalyze the insertion of a single oxygen atom from oxygen into an organic substrate. Okay. So this is a monooxygenase. That is, a single oxygen atom is being inserted into phenylalanine. As we've already compared the structures that the okay. I think we lost connection. Okay, so we were talking about the conversion of phenylalanine to tyrosine. So the enzyme involved in this irreversible reaction is your phenylalanine hydroxylase, which is a monooxygenase. That is, the enzyme is able to insert a single oxygen atom into the organic substrate. So we've already compared the structures of phenylalanine and tyrosine. There was only a difference of one hydroxyl. So what is happening? This is a hydroxylation reaction. So your phenylalanine, it changes into tyrosine. Now for this reaction, the enzyme is phenylalanine hydroxylase. Apart from that, what is needed? As you can see, tetrahydrobiopterin is needed, which is being converted into dihydrobiopterin. Okay, so your tetrahydrobiopterin changes into dihydrobiopterin, and also for this reaction, what is needed? The regeneration of tetrahydrobiopterin is needed so that this continues. So your dihydrobiopterin, it changes into tetrahydrobiopterin by the enzyme dihydrobiopterin reductase. And for this enzyme, what is needed? NADPH is needed. So that was about the conversion of phenylalanine into tyrosine. Now the question arises that where is this tetrahydrobiopterin coming from? So it is being generated from GTP. Okay, so your tetrahydrobiopterin, it is derived from GTP. Now from GTP, multiple enzymes are required. That is a multi-step process, and ultimately what is formed? Tetrahydrobiopterin is formed. Now one of the enzymes in this pathway is um, GTP. When tetrahydrobiopterin is being formed, one of the enzymes is GTP cyclohydroxylase. Okay. Now, talking about the clinical correlations from this one particular step. So there are three types of phenylketonuria. Phenylketonuria 1, which is called classic phenylketonuria, phenylketonuria 2 and 3, which is non-classic, and phenylketonuria 4 and 5, which again falls into the non-classic Phenylketonuria. So talking about classic phenylketonuria, it is very simple. That is, there is defect in phenylalanine hydroxylase. Now in type 2 and 3, there is defect in dihydrobiopterin reductase. So this is the enzyme which was converting dihydrobiopterin into tetrahydrobiopterin, as we've seen here. This enzyme here. This enzyme here, this is defective, okay? And your phenylketonuria type 4 and 5, there is defect in the formation of tetrahydrobiopterin. So as I told you, this tetrahydrobiopterin, this was formed from GTP. So there is defect in formation of tetrahydrobiopterin. So that falls under type 4 and 5. Now talking about the catabolic fate of phenylalanine and tyrosine. So your phenylalanine, it is changed into tyrosine. The enzyme is phenylalanine hydroxylase. So this is a hydroxylation reaction, as we've already discussed this step. Then this tyrosine, it is undergoing transamination. So the enzyme is tyrosine transaminase. Okay, tyrosine transaminase. This second step, it requires PLP, and what is formed? Para-hydroxyphenylpyruvic acid is formed. This is the keto acid of tyrosine. You can see here the diagram, although the structures are not required. Then what happens? This para-hydroxyphenylpyruvic acid, it is acted upon by para-hydroxyphenylpyruvate hydroxylase to form what? Homogentisic acid, also known as dihydroxyphenyl acetic acid. Then it is acted upon by homogentisic acid oxidase, another enzyme, to form what? To form malonyl acetoacetic acid. Then this malonyl acetoacetic acid, it is acted upon by malonyl acetoacetate isomerase to form fumaryl acetoacetic acid, and then this fumaryl acetoacetic acid, it splits into acetoacetic acid and fumaric acid. Okay, so your fumaric acid, it has glucogenic fate, and your acetoacetic acid, it has glucogenic fate. So this is why we say our phenylalanine and tyrosine, they are both glucogenic and ketogenic. And this is brought about by the enzyme fumaryl acetoacetate hydrolase. So that was about the catabolic fate of phenylalanine and tyrosine together. Now talking about the clinical correlations. So phenylketonuria, we've already discussed the first step when your phenylalanine was changing into tyrosine. So phenylalanine hydroxylase, that is defective and leads to phenylketonuria. Very important. Tyrosinemia, again very important. It is also known as tyrosinosis, and it is of 3 types. We will study about each of them in detail. And alkaptonuria, this is because of defect in homogentisic acid oxidase. Okay, so now let's take each of these clinical correlations separately. So talking about tyrosinemia type 1, which is hereditary, also known as hepatorenal tyrosinemia, and it resembles porphyria. Okay, now this is the most common tyrosinemia, and the peculiar manifestation of this is that there is cabbage-like body odor. Now the enzyme defective is fumaryl acetoacetate hydroxylase. So this was the last enzyme which was splitting your fumaryl acetic acid into acetoacetic acid and fumaric acid. Now type 2 tyrosinemia, it is also known as oculocutaneous or Richter-Hanhart syndrome. Now the enzyme defective is tyrosine transaminase here, and clinical manifestations include, they can be either it can be ocular manifestations or cutaneous. So in ocular, you can see corneal ulcers are there, and in cutaneous, there is non-pruritic hyperkeratotic plaque usually seen in soles and palms. That is what is pruritic? That is non-itching. Okay, no itching. Hyperkeratotic plaque seen in soles and palms. So these were the clinical manifestations of tyrosinemia type 2. So about type 1, the important thing is there is cabbage-like body odor, and the last enzyme, that is fumaryl acetoacetate hydroxylase, is defective. In tyrosinemia type 2, also it is oculocutaneous. The other name for it is oculocutaneous. Okay, so there is ocular manifestation, that is corneal ulcers, and cutaneous manifestations. Okay, Now moving on to type 3 tyrosinemia or neonatal tyrosinemia. So this is the least common tyrosinemia and the enzyme defective is para-hydroxyphenyl hydroxylase. That is defective. And if there is complete deficiency of para-hydroxyphenyl pyruvate hydroxylate, then type 3 tyrosinemia is caused. But in case there is not complete deficiency, okay, and only the enzyme is partially mutant, okay, so what is happening? It causes Hawkinsinuria. Now, the mutant para-hydroxyphenyl pyruvate hydroxylase, which is partially active, produces a compound Hawkinson. So, the peculiar manifestation is that there is swimming pool body odor. So, what is it trying to say? The enzyme defective is your PHPP hydroxylase. So what is happening? If it is completely defective, tyrosinemia type 3 is caused. But what if it is partially defective, okay? And it is partially active. So if it is partially active, it will lead to the formation of another compound which is known as Hawkinsinuria, and this Hawkinsinuria contributes to this swimming pool body odor. Now, talking about classic phenylketonuria, this is very important and very frequently asked in exams, so pay close attention to this. The enzyme defective, as we've already discussed, is phenylalanine hydroxylase. So, phenylalanine is accumulating. Now, this phenylalanine, it will enter alternate metabolic pathways. So, firstly, what will happen? Your phenylalanine, it will undergo transamination and form its corresponding keto acid, that is phenyl pyruvate. Now, this phenyl pyruvate, it can either undergo reduction to form phenyl lactate or it can either undergo decarboxylation to form phenyl acetate. Okay. Now, this phenyl acetate, it undergoes conjugation to form phenyl acetyl glutamine. And that phenyl acetate that is formed, it is responsible for the mousy odor, that is a peculiar clinical manifestation of phenylketonuria. So if there is mousy odor, it is because of phenyl acetate. And also, now one question arises that if phenyl alanine is accumulating because of defect in phenylalanine hydroxylase, then why it is not called phenylalanine and instead called phenylketonuria? Because we've already seen that phenylalanine that is accumulated actually enters other metabolic pathways, okay, and it is shunted to form phenyl pyruvate, which is a keto acid, okay, therefore it is known as phenylketonuria. Okay. Moving forward, it says that as phenylalanine is not converted to tyrosine, leading to decreased tyrosine, which in turn means there's decreased melanin, there's decreased catecholamines, and decreased thyroxine, because these are the anabolic products of your tyrosine. Okay, so once the tyrosine is not formed, it is very obvious that melanin, catecholamines, and thyroxine, they will also not be formed. So because of decreased melanin, what will happen? There will be hypopigmentation and it will be manifested as blue eyes, blonde hair, and fair skin. But there might be some dietary tyrosine. Okay, through diet, you might be getting some tyrosine, which might contribute to a little amount of pigmentation. So there is not complete absence of pigment because through diet, you're still having some tyrosine. Now, decreased catecholamines, that there is decreased neurotransmitters leading to neurological manifestations like intellectual deficits, agitation, and hyperactivity. Now, moving on to the laboratory diagnosis of phenylketonuria, you have Guthrie's test and ferric chloride test. What is happening in Guthrie's bacterial inhibition test? There is a bacteria called Bacillus subtilis, which needs phenylalanine for its growth. So if there's a lot of phenylalanine, so there will be many cultures of this bacteria, that is Bacillus subtilis, which means that there is defective phenylalanine hydroxylase enzyme. That is why you have excess or accumulating phenylalanine. So the amount of colonies formed is proportional to the amount of phenylalanine in blood, and the sample that is taken is usually through heel prick. Now the other test, that is ferric chloride test, In this, what is happening? To 1 ml of urine, you add ferric chloride reagent and a transient blue-green color is formed. This test is answered by phenyl pyruvate. That is, for a few minutes, a blue-green color is formed if you add to 1 ml of urine ferric chloride reagent. And what is the compound that is answering this test? That is your phenyl pyruvate. Okay, so this is not answered by phenylalanine, but rather given by phenyl pyruvate. Now moving on to alkaptonuria. Now, this alkaptonuria, it is part of Geroid's tetrad. What is Geroid's tetrad? It was studied by Archibald Geroid, and 4 diseases, that is 4 inborn errors of metabolism that are included in this tetrad. They are cystinuria, alkaptonuria, albinism, and pentosuria. You can try to remember this through this mnemonic known as CAP. Okay, this gap here. So this is a great way of learning. So you have cystinuria, alkaptonuria, albinism, and pentosuria. Now enzyme defective is your homogenesic acid oxidase. So your homogenesic acid, which is formed, it accumulates and forms benzoquinone acetate, which then polymerizes to form black-colored alkaptone bodies. So by the 3rd or the 4th decade of life, the patient may develop ochronosis, which is deposition of these alkaptone bodies in cartilaginous tissues like your intervertebral disc, nose, pinna, sclera, etc. Okay. So what is happening? Your your parahydroxyphenyl pyruvate, parahydroxy. I'm trying if I can write with my finger. Oh, actually, very bad. Okay, it forms homogentisate. Okay, now this homogentisate, it will not be able to form what? Malonyl acetoacetic acid. Okay, because the block is here. So what will happen? Your homogenesic acid oxidase. This enzyme is defective. The block is here. So your homogentisate, this will accumulate. Now, when this accumulate, what happens? This forms benzoquinone. Benzoquinone acetate. Now this polymerizes to form your alkaptone bodies. Okay. Now these alkaptone bodies that are formed, they can get deposited in cartilaginous tissues like in between the intervertebral disc, your sclera, your pinna and nose and all that. Okay. Now moving on to diagnosis. The urine becomes black on standing due to oxidation of homogenesic acid. Okay, so you can see 2 test tubes are there. So what is happening? There is excretion of homogenesate in the urine. So freshly taken urine, freshly excreted urine, it will be normal in color. But what happens on standing? It becomes black because of oxidation of homogenesic acid that is present in it. Therefore, this is also known as black urine disease. So you can see here in this picture, the specimen on the left, which has been standing for 15 minutes, shows some darkening at the surface due to oxidation of homogenesic acid. So it is beginning from the surface surface. Okay, so the oxidation is taking place from the surface. Now you can see after 2 hours of standing, the entire test tube has turned black. And also in the picture next to it, you can see deposition of alkaptone bodies in between intervertebral discs. You can see these black color discoloration here. These. Okay. Now moving on to the anabolic fate of tyrosine. So first is catecholamines, then we spoke about melanin and thyroxine. So we'll take catecholamines first. They include epinephrine, norepinephrine, and dopamine. They are produced by adrenal, medulla, and sympathetic ganglion. So talking about the synthesis and degradation, although the cycle is not very important, but you should know the first enzyme at least. That is from tyrosine. Another hydroxylation. Your tyrosine was formed by a hydroxylation from phenylalanine. Okay, now another hydroxylation, as you can see 2 hydroxyls here. Another hydroxylation takes place, and what is formed? Your dopa is formed. Now this dopa, it forms dopamine. From that dopamine, norepinephrine is formed, and from that norepinephrine, what is formed? Your epinephrine is formed. Okay. Just a second. Okay. So your tyrosine, it is acted upon by an enzyme known as tyrosine hydroxylase to form DOPA. DOPA is your dihydroxyphenylalanine. So tyrosine, there was already one hydroxyl. Now another hydroxylation is taking place to form a dihydroxy. Now this dihydroxy phenylalanine just acted upon by DOPA decarboxylase, that is decarboxylation is taking place. You can see a carbon dioxide molecule is removed and what is formed? Dopamine is formed. From that dopamine, you have your norepinephrine and epinephrine being formed. And in case of degradation, your DOPA is degraded to homovenilic acid, and your norepinephrine, it is degraded to normetanephrine, and then it finally forms vanillic manzanic acid. So that is briefly about the synthesis and degradation of catecholamines. Very rare that you get asked about this cycle. But the first enzyme, that is tyrosine hydroxylase, which is involved in the synthesis of DOPA, that is important. Now moving on to melanin synthesis. It is synthesized in an organelle known as melanosome in melanocytes. It is important in providing pigmentation to your skin, hair, iris, and retina. Then again, if you see the pathway of melanin synthesis, you can see tyrosine is again forming DOPA. But here the enzyme is very different from the one which was used in the previous pathway, that is in case of catecholamines. So here your tyrosine is changing into DOPA and the enzyme involved is tyrosinase and not tyrosine hydroxylase. Although hydroxylation is taking place in both the reactions, DOPA is being formed, but the enzyme used is different. So we'll talk about that. What is the difference between the two enzymes? So your tyrosine, it changes into DOPA, then that DOPA forms dopaquinone, then indolquinone is formed, and then finally you have melanin from it. So the main thing is that you should know that catecholamines and melanin and thyroxine, they are the anabolic products of your which amino acid? That is tyrosine. Okay, so that is more important for you to know, and not exactly the cycle. So the clinical associated with this is albinism. That is in case of defect in tyrosinase enzyme, you what is caused? It causes albinism. That is, you have milky white skin and hair and red eye reflex and all that. So that is albinism. Now talking briefly about the difference between the tyrosine hydroxylase and tyrosinase enzyme. Now both are monooxygenase and they convert tyrosine into DOPA. So this is the similarity. We've already seen how your tyrosine via hydroxylation is being converted into DOPA. And both of these enzymes, they are monooxygenase. That is, they are involved in the incorporation of a single oxygen atom into the organic substrate. Now, what is the difference? Tyrosine hydroxylase, it is expressed in those sites where catecholamines are synthesized, and your tyrosinase, it is expressed in sites where melanin is synthesized. So that is the difference, okay? That is, your tyrosine hydroxylase is expressed in sites where catecholamines is required, and your tyrosinase is expressed in melanin synthesizing sites. So therefore, a person with albinism, that is where your tyrosinase is defective, will still have normal levels of catecholamines, because your catecholamines, they are not dependent on tyrosinase enzyme. They are rather dependent on tyrosine hydroxylase. So that is the reason that even in an albino, your catecholamine levels will be same. So this forms an important question, okay? Now moving on to thyroxine. So thyroid follicles, they contain a protein which is known as thyroglobulin, which has 115 tyrosine residues. These tyrosine residues, they get iodinated. So what is happening? In your thyroid follicles, there is a protein which is known as thyroglobulin. It has tyrosine residues, and when these tyrosine residues, they get iodinated, they form what? They form MIT and DIT, that is monoiodothyroxine and diiodothyroxine. Now, one monoiodothyroxine, it might combine with another diiodothyroxine to form T3, or two diiodothyroxine, they might combine together to form T4. So that is how your thyroxine is formed. So that was all about phenylalanine and tyrosine, and we've already taken urea cycle. So with this, we come to the end of today's lecture. So this was very important both your urea cycle reactions and its disorders, and in case of aromatic amino acids, at least the pathway. Sometimes you might get asked about it. Then it's anabolic fate, and especially all the disorders associated with them. That is phenylalanine, very, very important. Alkaptonuria, tyrosinemias, all are very important. Okay. And that's it. With this, we come to the end of the class. And my pen is not working. Okay. Anyways, so if you have any queries, you can send them in. So see you tomorrow in the next class, and we'll be talking about tryptophan, glycine, serine, and any more amino acids if we're able to cover. Okay. Okay. Thank you for watching. See you tomorrow.